Morning, everyone. I'm just gonna wait like 3, 4 minutes just to let people in, and then I'll start.
Good morning. Okay, everyone, let's start. It's 8:04. So before starting, let's say this lecture, first of all, I want to let you guys, I hope everyone's safe, that you and your loved ones are safe and are doing well. I know this is like what, my second time in LAU, and every single time I come to LAU to teach, I get stuck in the war. So, مش جناحس. What can I say? Sorry about that. But no, I hope everyone is doing fine. And I know it's quite hard. I can only imagine how much what you guys are going through. I know this is your last year, and you're trying to get your perfect GPA or whatever, try to think about the future, and now you need to basically study and everything in the middle of a war. It's not the easiest. I really know it's not the easiest thing. The only thing I can really say, and this is easier said than done, it's try to use your classes or this work as an escape. For me, the best thing for me to escape the news, to escape the bombs and everything around me is work, for me personally. because I just need to focus on something and not focus on the outside. And I hope you guys can do that. That being said, if you guys think, this is too much, the war and doing keeping up with the work, with this course, with a lot of stuff, please, please, please be, my door is more than open to anyone. You guys have my email, you guys have my WhatsApp, more than welcome to text me, email me, whatever, and we can basically figure out something. Either, you know, I can help, or then I basically will ask someone from the LAU board to come and help. Basically, be it a therapist or be it someone from Gold in the faculty. So please, there's no technically, let's say, this kind of basically my office hours were not from 9, it was 9:30 10:30, something like that every day. That doesn't exist anymore. It's just now basically whenever when I'm available, email me, send me a message. It's faster, and then I'll definitely hop on Zoom or basically WebEx or wherever I can. So do use that uh and let me know. So basically I wanted this Thursday to be basically a pub quiz kind of thing, but because we postponed the exam, I didn't see the necessity for it because I saw that the majority of the people wanted it to be closer to the exam, and we've completely postponed the exam. It's not going to be this Saturday. Once I will basically, I still don't know when to postpone the exam. Right now, I'm just wanting for you guys to get used to this whole thing, and then we'll start thinking about the exams and stuff like that. But definitely, please do also take into consideration that I know what you guys are going through, so I'm not going to postpone the exam super close, and I'm not gonna make the exam super difficult where you already guys are in a very difficult situation. So I'll definitely keep that in mind. So I'm going to quickly now start today's lecture. So today I'm going to start with the last basically chapter. I'm going to also turn off my quickly just my camera just for the sake of internet. Okay. So can I quickly just get someone to tell me if they can see my screen,
I
like the
can see it.
presentation? Perfect. Thank you. Thank you. All right, everyone. So let's basically the last time basically we were discussing, which was last Thursday, we were talking a lot about autoimmunity and tolerance. Specifically, basically, how do we teach our own immune system, specifically our B cells and our T cells, not to kill us? We're teaching them not to recognize self-antigens. And this is extremely important because once they start to recognize our own body, that's when we start to get autoimmune diseases. And there's not just one type, there's so many types of autoimmune diseases that are out there. And as I said, understanding what factors contribute to autoimmune diseases are quite of a black box, honestly. We cannot really say what it is sometimes. We know, of course, certain diseases are quite simple, but some of them are quite complex. And today we're going to dive into that. That's quite cool, and I'll definitely be giving you some nice cases that we've seen. But also, we'll be talking about transplantation because transplantation can be, in some sort of way, a quite similar to autoimmunity, and they go hand in hand. So they're always usually in immunology, they're always taught together, and we'll definitely be talking about that why. So, as I said, autoimmunity or autoimmune diseases, it's quite complex. It's not just one thing. It's not that it's a genetic factor or a environmental factor that by itself can cause it. Sometimes it is. Sometimes it is as simple as that. But in most of the cases, it's not. It's usually a mixture of having a genetic predisposition, meaning that you have a certain mutations in one or more than one gene that makes your immune system much more receptible or more susceptible to recognizing self-antigens, and then an environment or a cue, like an exposure, be it an infection or something in the environment that pushes that genetic factor to cause this immune regulation. And basically, you're causing your B and T cells to start recognizing self-antigens, and this induces autoimmunity. So we're gonna talk about different types of factors today. And one of them is a genetic component. And we really started to understand this genetic component quite a lot when we started to see a lot of families, per se, having similar genetic diseases being passed on from one generation to the other. So that clearly is an indication of, inheritance, that there's a genetic component towards it. But we also started to see this a lot in mice. So in mice, specifically in mice that we do like kind of this experimentation with, you have something called mouse strains. Every mouse strain is exactly genetically identical. So if you have, for example, in this case, the notch strain, that means that all of these mice in this notch strain are genetically identical. They are inbred. They have literally the same genes. So we have the white mice, which we call them bulb C mice. These bulb C mice have a genetically identical genes. Then you have, for example, the black mice, which we call them the C56 black mice. Those mice are genetically identical with each other. And one thing that we see that is quite interesting is that this kind of strain that we call the notch strain, this notch strain per se, for some reason, they almost all of them developed type 1 diabetes. For some reason, basically their incidence of diabetes increases. So here you have basically this not mice. The x-axis is the age over weeks, and the y is the incidence of diabetes. And you have in red the female, and then in blue the male. So one thing that you see is that first of all, over the age, you have an increase of basically diabetes, which you don't see with other mice. You don't see this with the white bulb C mice or the black C56 black mice. You don't see that. You only see this with only node kind of mice. So this again tells you that, hey, there's a genetic component towards this. But another reason or another, let's say, evidence that there's a genetic component, look at the female mice compared to the male. There's a big difference. The female mice are, especially at, let's say, week 20 or at 18, 60% of basically of the female mice have diabetes, while in the male, there's around like 22, 23%. So you see that for some reason, females are much more prone to type 1 diabetes in the non mice. Well, this is not just in mice. This is also in humans. In humans, if you basically go see the prevalence of autoimmune diseases when it comes to males versus females, you get to see that females are much more predisposed to autoimmune diseases. For some reason, this is still a black box to us still. We have some sort of basically explanations, but it's quite hard. We're still getting a lot of research of why females are more predisposed to autoimmune diseases. But of course, the common denominator here is the X and Y linked genes. That the X genes basically have a lot of extra, let's say, immune related genes that are not found in the Y. The Y, we know that there's very little genes to nothing almost there compared to an X. So that's why females basically are more predisposed. One reason. So this basically shows you that first of all, not this specific not strain mice are predisposed to type 1 diabetes because of the genetics, but also basically the female and male basically differences in why is female having much more compared to male? Well, this is more in the genes. So this shows you a genetic component per se. But one thing that is also interesting here that other than just tells you a genetic component is involved. Why is this, for example, mouse over here? So if we look at basically the male mouse over here, why is this male mouse getting type 1 diabetes here at week 14 and this one getting at that week 30? Why is that so? And the same with the female, why is this one getting it at week 13 and the other one's getting at week 30? They have the exact same genetics. This technically, if let's say autoimmune diseases are just purely genetics, then we should see a straight line, not basically an increase. This should be basically them all spontaneously getting type 1 diabetes at the same time. But we don't see that. We see some mice getting it at weeks 13 and the others at week 30, for example. This tells you that there's an environment influence, that something in the environment at week 13 induced this mouse to get it, and this one at week 30, basically, they got it later because of certain influence. So this graph, I love it because it gives you kind of the indication of two things, a genetic and an environmental influence, in a way that there's clearly a genetic influence, both in the female versus male, but also in the notch strain, but also then an environmental influence because they have the exact same genetics. Why is one getting it at week 13 and the other one at week 30? This kind of helps you understand that, hey, there's something wrong here. Like there's something in an environmental cue that's pushing the genetic factor. And we'll be talking a lot about these environmental factors, which to my opinion are extremely interesting. So of course, after we started to learn how to do genetic, basically sequencing and specifically genomes, our understanding and etiology, or basically kind of understanding of mechanisms of diseases, just exploded. Back in the day, it took us literally a fucking decades to actually genome a whole sequence. Well, nowadays, I think I read there was a breakthrough that now I think sequencing a whole genome, exome wise, costs you around $100. Before it would cost you millions and a decade. Now it takes a week and $100. That's pennies. That's literally pennies compared to before. So honestly, we are getting so much genetic information that not just autoimmunity is being undercovered, basically. We are having a literally massive surgence of kind of, let's say, mechanistic data that right now we have so much genetic data that we don't know what to do with it. We are trying to actually understand, basically try to understand much more complex diseases than before. And an example here is this, for example, I'll give you an example of how much genetic data has been able to give us. This is a beautiful umbrella. These are called umbrella plots because they literally look like umbrellas. So every single big dot over here that's colored is an autoimmune disease. So this is systemic lupus erythematosus, psoriasis, basically inflammatory bowel disease, type 1 diabetes, rheumatoid arthritis, multiple sclerosis, all these different types of autoimmune diseases. And every single small little blue, basically, dot that's connected is a gene and basically its connection towards what autoimmune diseases it's linked. So, for example, you have HLA-C. HLA-C, that is this one gene that is basically, if something goes wrong here, you have psoriasis. The same thing goes, for example, with HLA-DRB1. It's connected to MS. So it tells you that, hey, if you have something wrong in HLA-DRB1, you have most probably going to have some MS susceptibility. So you get to see many different types of genes that are connected to different autoimmune diseases. But one thing that we also saw that was really interesting for everyone was that it's not just like one gene, one autoimmune disease. Sometimes, for example, in the case of here, this TNF alpha gene, it's connected to literally four, to lupus, psoriasis, IBD, and rheumatoid arthritis. So you start to see that there are certain risk genes for one autoimmune diseases, but there are also some that are for multiple. So we start to see that, hey, there is basically genes that are really central to many autoimmune diseases. And these are different single gene traits, and I want you guys to know these because these are all things that we've went through, and they should be quite easy for you guys to kind of, let's say, memorize or understand. The first example is IRE. If you guys remember IRE is the complex that helps mesenchymal thymic epithelial cells to basically present these tissue-restricted antigens. So they help basically the negative selection. So it helps this mesenchymal cells to present self-antigens, a variety of different tissue-restricted antigens towards your T cells. And of course, if the T cell is recognizing it, then it's negatively selected. It means it kills it. If you knock this out, if you knock this IRE out, basically in a mutant, or you have a mutation in it in humans, you have a disease called APS. It's a syndrome, and it's a syndrome in where you have not just one autoimmune disease, but you have an amplitude of autoimmune diseases. You have all different types because here it depends on whatever T cell comes out because you don't have negative selection and T cells now this whole, let's say, barrier that is not allowing self-reactive T cells to be killed is gone. So now they're basically being unleashed and going everywhere to kill. So basically, this is one gene that can cause autoimmunity, multiple types. CTLA4. CTLA4, we talked about CTLA4. This is extremely important for the peripheral tolerance. Basically, this is actually used by Treg cells to stop, basically, or to induce anergy. So CTLA4 can basically bind to the dendritic cells to stop the presentation of any self-antigens, but also can basically stop other T cells to go into anergy. And if you knock this out, you get Graves' disease, you get type 1 diabetes, you get rheumatoid arthritis, you get many different types. So CTLA4 is basically a central, extremely important protein. And basically that is really needed for peripheral tolerance. Another example is FOXP3. FOXP3 is the transcription factor that induces CD4 T cells to become Tregs. You need them. We know that Tregs are an extremely important cell when it comes to the peripheral tolerance. Without the Tregs, what cell is going to stop your immune system to basically to go into resolution? So Tregs are extremely important. You get IPEX, this disease, which is almost exact same as APS, and where you have multiple different types of autoimmune diseases. Seriously, your body is just going haywire with autoimmune diseases. Then you have FAS. If you guys remember FAS is basically the death receptor. Remember in B or T cells, when they actually bind extremely high at high affinity for T cells or high avidity for B cells, this kind of is a signal that, hey, this is not normal. This is most probably a self-antigen because it's too strong. And you basically upregulate the FAS receptors, these death receptors. And this basically helps the apoptosis of self-reactive B and T cells. While you remove it, you basically induce ALPS, which is exactly the same as APS. It's basically this holy autoimmune disease syndrome. You're just getting so many autoimmune diseases. Another one is C1q. C1q, we spoke about this. This is a extremely important protein, a very important protein that is needed for your classical activation of your complement cascade. So your complement system, these signaling proteins that are being cut to create this membrane attack complex that is part of your innate immune system. So C1q is extremely important to activate the classical pathway. If this is removed, you're not allowing your body to remove apoptotic cells or basically immune complexes, because the C1q creates a lot of these kind of MAC complexes or basically C3b to opsonize these immune complexes or apoptotic cells to be cleared out, to be phagocytosed technically. If you don't have C1q, you don't have opsonization, you don't have MAC complexes, the apoptotic cells are not going to be cleared. These apoptotic cells are releasing self-antigens left and right. So you're really inducing, and specifically you're inducing systemic lupus erythematosus. So lupus is very connected to the C1q gene. And of course, this I don't want you to understand, to memorize, just to know that many different types of cytokines can be basically also basically induce technically autoimmune diseases like TNF alpha. Have too much of it can cause a huge basically inflammatory, let's say, response, which can basically lead to a lot of death of normal cells, reducing self-antigens. The basic removal basically of interleukin 10, for example. Interleukin 10 is the overexpression of interleukin 2, 7. These are basically very important cytokines in T cell activation, interleukin 3 as well. but also the underexpression of them. For example, TGF beta is a very important cytokine for Tregs. So this one helps to actually stop T cell activation. So you underexpress it, you're kind of letting T cells to overexpress. But one thing that you see interesting that caught my eye as well is here in the overexpression of TNF alpha. If you overexpress it, you get IBD and vasculitis. If you underexpress it, TNF alpha, you get lupus. But this is to show you that your immune system is not black and white. You don't have black and white shades of it. You have literally just gray areas. In a sense, what's trying to say is that it's not the sense of either it's present or not. No. Sometimes what happens here per se is that you have cases in where, in the case of in this overexpression of TNF alpha, too much TNF alpha is actually causing your immune system to kill so many, normal cells. And the killing of normal cells releases a lot of self-antigens, which can cause IBD, arthritis, vasculitis, you name it. But in this case, in the underexpression of TNF alpha, well, TNF alpha is also extremely important to help T cells remove basically damaged cells, or help basically macrophages come in and remove apoptotic bodies. If you have an underexpression, you're not allowing that. You're not allowing the removal of apoptotic bodies that are like cells that are damaged, and you get a lot of release of antigens. So the immune system is delicate. It's in a sense, you don't want to overexpress it and you don't want to underexpress it as well. You want a fine balance of it. And finding that fine balance sometimes can be extremely tricky. And this is why autoimmunity, if you look at basically a lot of diseases are connected to your immune system, because it can mess up quite easily, sadly. But of course, as I said, we started to do a lot of we started to gather a lot of genomic data from human beings. So we started to do these kind of certain studies that we called genome-wide association studies, or in short, GWAS. So what a GWAS is, technically, is you're looking for a correlation between the frequency of how much you get a disease with genetic variants. And specifically, the genetic variants, we're talking about SNPs, single nucleotide polymorphisms. This means one nucleotide differences. So that's what it is. So we're trying to see, can we basically locate SNPs in human beings? And can we see a correlation with the frequency of a certain disease? So I'll give you here a nice example of this. This is a GWAS basically plot, or we also call this a Manhattan plot, which tried basically to link certain mutations, so different SNPs in different chromosomes, with Crohn's disease. So Crohn's disease is another type of autoimmunity. It's a very horrible, basically, kind of disease coming to your intestine. So, for example, here you see in chromosome number 1 that you have this SNP here in gene interleukin 23 in chromosome number 1, that if you have a SNP here, you get a basically almost 60% basically association with Crohn's disease, and you have all these different types of basically genes. So this GWAS kind of study really started to help us understand all these different genes. This is what basically told us, like, hey, these genes, like having SNPs, these single nucleotide polymorphisms, these single nucleotide differences, can cause or give us susceptibility to all these different types of autoimmune diseases. And we really started to see really a lot of patterns. For example, one thing that we started to see is that this ATGL16L1, this gene is CTLA4. Well, we started to see here that CTLA4 is causing Graves' disease. If you have SNPs here, it can give you predisposition to Crohn's disease. But not just that. You disrupt that CTLA4, you get type 1, you get Graves' disease, Hashimoto's, rheumatoid arthritis, MS, you name it. Because as we know, regular T cells, they stop basically self-reactive T cells, they put them into anarchy or suppress them, or they basically kind of stop also dendritic cells, basically, because Tregs use CTLA4 in abundance. you mess that up, you mess up your peripheral tolerance. So this is where GWAS studies really helped us to kind of identify very important genes for autoimmune diseases. But one thing that we started to see, for example, if you look at interleukin 23, we see basically also interleukin 12, card9, mucin 19, NOT2. All of these genes that I just mentioned, they are actually connected because card9 and this PTGR4, these guys are PRRs. They are pathogen recognition receptors that they basically will recognize a PAM, a pathogen-associated molecule. For example, beta-glucan. This is coming from bacteria or fungus, and this activates your PRRs, and this can activate your card9 or PTR3, which causes the dendritic cell to produce a lot of interleukin 12 and 23. 12 and 23, what they do is they bind to T cells, CD4 T cells, and they help literally the production in T helper 1 cells and T helper 17 cells of this interferon gamma, interleukin 7, 22. So technically, dendritic cells, once they're activated, interleukin 12 is being released. It's binding to T helper 1 cells. They're making them activated to produce interferon gamma. Interleukin 23 is basically binding to T helper 17 and pushing T helper 17 to release these 17 and 22 interleukins. So you see that this, if you have SNPs here in the interleukin 23 receptor, 12, card9, mucin, NOD2, which are all found basically card9. This is your basically PGR34. Basically, you have also this interleukin 12 receptor, 23. If you have SNPs in these genes that make them suddenly have a abundant function, so a gain of function, that means that now these receptors are actually much more active. You're pushing your T helper 17 and T helper 1 cells extremely towards the brink. They're being overactivated. Having overactive T cells, well, those are the ones that can cause basically autoimmunity. So you're giving yourself a predisposition to these autoimmune diseases. And this is where the GWAS kind of studies helped us. So this GWAS kind of studies, this is this one example, but we've done them for basically almost every single type of autoimmune disease possible. And this gave us categories. So we started to see genes basically that are grouped in affecting the autoantigen availability and clearance. So the autoantigen availability, this is, for example, IRE. The IRE gene basically is extremely important for presenting these TRAs, these tissue-restricted antigens. So they help you with negative selection. Or in the autoantigen clearance, for example, the C1q. Basically, if we don't actually able to clear autoantigens, we are not there. There's gonna be a higher chance of these autoantigens for them to be recognized by self-reactive T cells or B cells. So if you remove C1q, you reduce the clearance of it. Apoptosis, a lot of death receptors like fast receptors, if you remove those receptors, you are preventing self-reactive B or T cells to be killed. Signaling thresholds, cytokine expression or signaling, all these things go into the overactivation of T cells or underactivation even sometimes as well. Co-stimulatory molecules, as we said, CTLA4 is a co-stimulatory molecule. It's a negative co-stimulatory molecule that Tregs use to regulate basically these self-reactive B and T cells. So you see where we started to categorize them into them. And another one of these one is B7 or B7 or CD28. If you have too much B7 or too much CD28, specifically CD28, you're pushing your T cells to get overactivated. Regulatory T cells, you mess up your Tregs, you mess up everything. So any gene that messes up with your Tregs basically can cause autoimmune diseases. So we started to see a lot of different genes and you can categorize them into different, let's say, pockets, technically. So basically, as I said at the beginning, that sometimes you actually have certain, basically, autoimmune diseases that are, well, let's say, majority of autoimmune diseases are a combination of more than one gene. So if you go back to this GWAS, you see that Crohn's disease here, it's multiple genes. It's multiple genes that can be correlated with it. And that's the case for many different types of autoimmune diseases, and where you have more than one gene of being basically affected. But in some cases, you have monogenic effects. So we call these basically monogenic autoimmune diseases, and where one gene can cause it. And these are the ones that I spoke about. The IRA, basically, the CTLA4, the FOXP3, the FAST, the C1q, extremely important genes that are really affecting not just one type of cell, but affecting so many different, let's say, pathways. In the IRA, you're affecting literally the whole negative selection pathway. CTLA4, you're affecting the whole Treg, so you're affecting the whole peripheral tolerance. FOXP3, you're wiping out the peripheral tolerance, basically, almost by removing Tregs. So fast, you're removing the ability of B and T cells. So this is where you see central genes that are so crucial and important for tolerance that if you basically remove them, this can cause autoimmune diseases. So you have exceptions. You have certain monogenic autoimmune diseases, but in majority of the cases, it's mostly a combination of multiple genes. One gene that is extremely underrepresented and super important for you guys to know is MHC. MHC, as we know, is a very important gene or protein that helps basically our cells to present antigens to B and T cells, basically. We need them. They're the ones that present antigens, and specifically the T cells. So as we know, we have MHC2 and MHC1. Forget about this 3. This is something else. Or this is more for TNF alpha and beta. You don't need to know this. But of course, I need you guys to know that in the MHC2 and there's MHC1, as we studied before. So we know that with MHC1, this is mostly for endogenous antigens, so more for viral antigens. Basically, MHC2 is more for exogenous antigens like bacteria or fungus. But one thing that we also know, for example, in the MHC1 is made of a alpha, basically protein and a beta-2 microglobulin. So alpha chain and a beta-2 microglobulin make your MHC1. So in MHC1, you have 3 genes. You have MHC, you have HLA-B, HLA-C, and HLA-A. So HLA-A, C, and B make up MHC1. So they all encode, these 3 genes encode for all the proteins to create MHC1. But this is extremely polymorphic, in meaning that we have 8,000 different HLA alleles. You have 9,000 different, basically, HLA-B alleles, and you have 6,000, basically, HLA-C alleles in the humans. And you get 1 copy, basically, from your parent, 2 copies of HLA from your mother and your father, 2 copies of HLA from your mother and father, and 2 copies of HLA-C from your mother and father. So this, you get combinations. So imagine that you get HLA, basically here 8,000, basically 7,200, and then HLA basically 2,200, and HLA-C 2. Then that gives you basically an MHC1 that is different to the other. So you get different combinations of MHC1. Well, that actually different MHCs, what we've seen is that different types of MHCs with the different alleles has a different affinity to different antigens. We started to see that if you have a certain MHC, your MHC might actually bind much better to self-antigens. So they're linked to autoimmune diseases because in the heart of an autoimmune disease, it's all about the antigen. It's all about that self-antigen. And if you have an MHC that is able to bind at a high affinity to a self-antigen, you're increasing the risk of it to actually activate self-reactive B or T cells. And that's what's happening. And the same thing goes to MHC2. And MHC2, basically, it's made of DP, DQ, and DR, basically, genes. So 3 genes that make up your MHC2. And there's so many different alleles. So you get so many different combinations. And we see that certain combinations of HLAs actually predispose you to have higher affinities. So if you go back to this basically umbrella plot, this is why we see so many HLAs. HLA-C, having a certain HLA-C predisposes you to psoriasis. Having this HLA-DRB1, basically this specific HLA can cause you multiple types of autoimmune diseases because it increases the affinity to so many autoantigens or self-antigens. You basically see another HLA, this is another HLA here. We start to see that HLA genes are extremely important. And this is extremely important, actually, if you know what HLA or what MHC means. MHC is major histocompatibility protein. It's a compatibility gene, meaning that no one has the exact same MHC. It's almost impossible. It's very hard to get to people that have identical, because look how many different types of alleles you get. The combination is immense that you can get of these. If you get to get MHC1, look how many different HLA, B, and Cs you have. So we started to see that certain people that have, for example, HLA 7221 has a predisposition, or people that have HLA DRB1 number 33,600 gives you a predisposition. We started to see this basically. So these are all different ones. I don't want you to memorize that D3. No, please no. I just want you to know that the allele, the MHC allele, correlates to autoimmunity. That's all I need you to know. And we see this, of course, in the MHC1, for example, here. But if you look closely, you see this much, much more in the HLA2 genes, more in the basically DR, DQ, and DP genes. And we start to see that a lot of these MHC2 genes, if you have certain one of them, for example, if you have D3 or if you have D2, you name it, if you have any of these ones, you have an increased risk. For example, let's imagine this one. You have DR2. So you have basically this DR number 2. This gives you a 15.9 relative risk of getting Good-Pasteur syndrome, because this DR2 actually increases your MHC2 to bind to self-antigens related to Gut-Pasteur syndrome. Same thing goes with B27. If you have B number 27 in your HLA, you have the huge increase of getting ankylosing spondylitis, so 87.4%. It's crazy of an increase, because that specific HLA increases your MHC to bind to self-antigens related to ankylosing spondylitis. So this is just to show. But more interestingly, other than just having, let's say, MHC alleles that are increasing the binding affinity, we also see single nucleotide polymorphisms causing this increase even to go more. I'll give you here an example. So you have DR3 and DR4 alleles. See, these are coming from the MHC2. So the DR3 and DR4 alleles, these ones, if you have them, they increase your type 1 diabetes, because D3 and D4 alleles, what they do is they give you an MHC2 that actually binds to more self-antigens like insulin or beta, basically these beta cells in your pancreas. But what we started to see that in some people, specifically, so these DR3 and DR4 alleles, they give off basically your alpha chain and your beta chain of your MHC2. Specifically in your beta chain, some people have a SNP in position 57. Normally, in normal people, you have basically here this red amino acid that this is basically positioned for number 57. This in general is an arginine. No, sorry, this is not arginine. This is basically a aspartic acid, my bad. This is a aspartic acid. This aspartic acid creates a salt bridge with your basically arginine residue over here. This kind of helps you not to bind to any self-antigens related to type 1 diabetes. In certain people that have this DR3 and DR4 alleles and that have this mutation as well, they actually do not have this aspartic acid anymore. You have it switch to an alanine, for example, which is here in yellow. This alanine doesn't do anymore this salt bridge anymore. This now causes an increase of affinity to self-antigens. So you have two things here. You have A, certain alleles of an MHC, either coming from the DP, DQ, DR, or the HLA-B, C, and A. There's many types that increase the overall affinity of the MHC to a self-antigen, or you have SNPs that causes basically a huge increase. Does this make sense? Do you guys want me to repeat this? Please let me know or put it in the chat. All right. Now on to this, basically the more, let's say for me, interesting parts. And this is I think you guys will enjoy this part. Because this is more in the environmental factors, and they're extremely cool in my opinion. So one thing we started to see is in the environment, one of the biggest environmental factors that we start to see is the way you're born. If you're born through the C-section, you actually have a predisposition to autoimmune diseases. You have a higher predisposition compared to babies that actually were born through the vaginal delivery. So we started to do a lot of these different types of studies, and we really saw that if you are born through the C-section, you have an increased chance of getting an autoimmune disease. And the reason behind this is purely because of your microbiota, your bacteria inside your stomach or your intestine, more likely, sorry, not your stomach, but in your intestine. So what we see is that if you're born through your C-section delivery, you have a different composition of bacteria in your intestine. And this causes your immune cells to be extremely overreactive and to be reactive towards self-proteins. If you're born through the vaginal delivery, you have a different. So if you look at the different types, basically here, you have different types of bacteria. This one is actually helping your immune system to be more, let's say, not that reactive towards yourself. So other than just basically the way you're born, the type of basically the type of microbiota that you have is a factor. So what we start to see in people, one example is if you're born through C-section or vaginal delivery, but it also can be through diet. It could be basically through the environment that you're exposed to. So many things can change your microbiota. It's crazy. If you exercise, if you're basically a very nervous person, the factors of it is alarmingly high. There's so many different things. But one thing that we see is in your gut microbiota, there's thousands of different families of bacteria there. And you can divide these basically families of bacteria into two different types. There is the first type that we call them the beneficial bacteria. So their name says it, they're beneficial. They help us basically kind of live a healthier lifestyle. And then you have the harmful pathogens or pathobionts, we call them as well. So these harmful pathogens, what they do is they create a huge inflammatory environment, a very pro-inflammatory environment in where we damage the gut. So our gut lining becomes leaky because you've damaged it. You destroyed so many cells that things are now leaky. So what happens is you have a lot of bacteria being released, a lot of stuff that shouldn't be released into your blood because your gut now is leaky. Well, what happens if this is very leaky? Certain bacteria can actually leave and they can cause autoimmune diseases. And one way of causing autoimmune diseases is through something called molecular mimicry, meaning that the bacteria per se can look so similar to a self-antigen. And we'll be coming back to this in a bit. So what happens is that your immune system starts to attack this bacteria that looks so similar to your self-antigens that your T cells, once they're attacking it, they will also attack the self-antigens, and this causes an autoimmune disease. but I'll come back to this later. You can also have an imbalanced flora. So what happens is that pro-inflammatory environment, what's causing it is causing your T helper 17 cells to actually be overactivated. Well, you overactivate T helper 17 cells, you're increasing the chances of self-reactivity because you're overriding T cells. You're making them proliferate so much that some of them can be self-reactive and they can be basically proliferating. And you're also reducing your Treg cells. Another thing is that beneficial bacteria, they release something called short chain fatty acids. And what short chain fatty acids do, they stop out to basically inflammation. Well, when you basically reduce the amount of beneficial bacteria and you increase your harmful pathogens, you reduce the amount of SVAs, so the short chain fatty acids, and you have just a huge auto basically kind of, let's say, activation of your immune system. So this is how your gut can really affect you. Of course, this can go all the way up to your nervous system as MS, Guillain-Barré syndrome, to your skin, to your joints, to your digestive tract, everywhere. It's connected to everything. Another very interesting, basically environmental factor is hygiene. There's the hygiene hypothesis. And this is basically what people started to see, is that people that are actually living in an extremely clean environment, so let's imagine people are living in a more industrialized area where it's more clean compared to someone that's living more in a farm area that's much more dirty and exposed to germs. Well, the person that actually lives in a clean area actually has a much more higher chance of getting a autoimmune disease. So epidemiological studies have shown this, that if you live in a very clean regions, you most probably are going to have higher rates of type 1 diabetes, Crohn's disease, MS, and you name it. Because what happens, what we start to see, if you are basically a child and you're basically surrounded by germs, of course, to a healthy manner. It's not that we're basically being, basically we are bathing in germs daily, but you're exposed to in a healthy manner to germs of different kinds. It actually helps us to become much more healthier. It makes our immune system, first of all, get taught how to kill these germs in a more sensible manner. And it also helps us to accumulate Treg cells over our lifetime. Because what we see with people that are actually living, let's say, grew up in industrial or cleaner regions, these people, they're not exposed to many actually bacteria or viruses or other pathogens. So what happens is that when they grow old, when they start to grow older, when they are exposed to certain microorganisms, the immune system is not so experienced that it starts to actually exaggerate in its activation. And an overactivation of T cells can induce autoimmunity. And these people do have actually a lower development of Treg cells because Treg cells come up after a infection. If you're a child that gets basically infected constantly, you're developing your Tregs a lot. If you're a child that is not exposed, your Tregs are not that developing. So this is where we see this. And this became such a huge headline that the Economic Times even wrote about it. Something that the Economic Times has to do something with purely with the economy of the world. They spoke about this hypothesis because it was such a huge kind of interesting thing for people. Another one is vitamin D. So what we see is people up in the north, like people living in Finland, like myself, sadly, Switzerland, Norway, basically Sweden, Norway, Denmark. These people actually have a huge increase in type 1 diabetes or MS. This is because they're not basically exposed to vitamin D. People that are living, for example, in the Mediterranean countries like Lebanon, Greece, Portugal, Spain, Italy, you name it, actually have a lower chance of getting type 1 diabetes or MS compared to Northern Europeans because they get vitamin D. And vitamin D has a huge role, actually, in stopping, literally, for example, in the case self-reactive B cells, and stopping dendritic cells and presenting self-antigens, and helping stopping T helper 17 cells. It does a lot of things. It has a very good kind of anti-inflammatory property that we really need. And for me, I take vitamin D pills literally starting September all the way to April because I barely see the sun, sadly, in Finland. And one last one that I want to talk about here quickly is your infection. That's your last, basically, kind of environmental factor. So, one thing that infections can do is they can actually cause so much death to our cells that can release so many antigens, a lot of release of self antigens. And of course, that increases the chance of self-reactive B or B cells to be activated. But it also causes T cells and B cells to increase co-stimulation. Basically, it enhances CD27 to be, sorry, CD28 to be upregulated. And this can cause self-reactive B or T cells to also start proliferating and creating more co-stimulation. They can also disrupt immune suppression because when you get infected by a virus or infected by a bacteria, well, you're just causing your immune system to go from an immunosuppressive state to a immune active state. So that can cause it. But the more interesting thing we see is a phenomenon called molecular mimicry. And I'll explain this using this amazing uh, figure. We have Streptococcus. Streptococcus, basically, when we're basically infected by Streptococcus, this bacteria, we create plasma cells that are, producing antibodies. You have two different antibodies. You have antibodies binding to this red triangle over here, shown here in yellow. And then you have these basically blue antibodies that are binding to this oval shaped green things. What happens is that a component in the streptococcus, and this is actually a true case, looks very similar to a component in our heart. So this component in our heart is this basically orange triangle. They are so similar in molecular structure that what happens is that these antibodies that are now produced, when they go into the heart, they will bind to your heart as well because they're so similar. The antibody gets confused and they start to bind basically here to these different types. So to your streptococcus wall and to your heart because they're molecular so similar in structure. And we see this a lot, not just for streptococcus, but for many things. I'm not going to go over this. This will not come in the exam, but I've recorded this and I put this. This is a very interesting case. I'm going to quickly go through this, but super quickly. We started to see this in China when the 2009 pandemic of H1N1 virus started to hit. We started to see that this is the narcolepsy. So narcolepsy is a disease, is a type of disease that makes you sleep a lot during the daytime. We started to see that, this is the cases in China throughout the years. You get like what, five, maximum 35 cases a year. In 2010, that went to 201. And people were surprised, like, why is this happening? Well, bit by bit, we started to see that basically, well, I'm not going to go through this again because this will not come in the exam. This is for your own case. I've recorded this, but what we started to see is that the H1N1 virus has two proteins. This basically neuraminidase Na and nuclear protein MP that look extremely similar towards proteins found in your brain. So what was happening were that your T cells that were recognizing neuraminidase and nuclear protein that now are expanded were going to your brain and they were literally killing basically certain receptors and causing you to have narcolepsy. So this was a very real case in 2009 that people that were actually infected with these influenza viruses were actually having narcolepsy because their T cells were going and actually killing certain receptors in your brain that was inducing narcolepsy. So guys, I'm going to give you literally 5 minutes of a break. So now it's 8:59. So let's say at 9:04, I'll come back and I'll quickly go through transplantation. It's a very couple of slides.
Uh, Doctor, do you hear me?
Yes, I can hear you.
Yes, Doctor, I want to tell you that I'm sorry, but I joined late today. I want to ask if I missed like anything of importance, like.
No, don't worry about it. You've not missed a lot. Basically, don't worry, and everything is I'm recording it twice. Like I recorded
Yes,
before and
yes.
after, so it's gonna be all online. But basically, it's just to tell you that hey, if there's anything I can help, because I know it's a stressful times, please let me know. All my office hours are now basically whenever you want. So send me an email or basically text me to tell me like, hey, let's have a meeting. I have some questions or have some issues, so it's all basically this policy of like 9:30 to 10:30 of my office hours. It's now basically free. So whenever you want
Yeah.
me, let me know.
Thank you, Doctor. Doctor, also I want to ask, what will happen with the exam on Saturday?
So for the exam sake, it's now postponed. There's no definite exam this Saturday. I don't know when I'm gonna postpone it. I don't want you guys to think about this right now. Neither like I think I want us to get used to, well, used to in quotations, big quotations, to hold this whole thing. I will basically will give you guys. I will let you at least a week or something in advance to tell you when the exam is. So I've not decided when to do the exams. Then this also comes to the provost, because as you saw, the provost does not allow any exams this week with a good
Oh,
reason for not to do so. So once they start allowing the exams, then I will plan my exam.
thank you, Doctor. Thank you so
No
much.
worries. No worries, guys. Okay, so it's 9:04, everyone. Honestly, guys, I am gonna stop here. I don't wanna, I'm not gonna, I feel I don't wanna over push this already with the current situation. I'm gonna stop here. Is there any questions that you guys have related to this core, related to this lecture today, or anything in general? All right. Well, I guess there's not questions or anything. One second, sorry. All right, perfect. All right, guys. Please take this time. Transplantation. I'll go over it. I want it to be more fun because I basically added a couple of extra slides about the new types of transplantations. I'll go over this later, or most probably on Tuesday or something with some other stuff. But yeah, everyone, please stay safe. If there's anything you guys need, please feel free to reach out to me. If not, great guys, have a great continuation of your day. Thank you, guys. Thank you.
Thank you, Doctor.
Thank you, guys. Have a nice one.